So we talked about mitosis. Mitosis is just a process by which cells increase in number and divide and grow and then divide and grow. And we talked about the cell cycle, where most of the cell's life is spent in interphase, and then it goes into a dividing phase. And not all cells do that, right? Some cells, like nerve cells in the brain, don't divide. Once they're formed, they're done dividing. Muscle cells and fat cells, once you're done growing and done with puberty, those cells typically don't divide. They don't undergo mitosis. But other cells do undergo mitosis. And what do we call that uncontrolled mitosis of cells dividing out of control? Cancer. Yeah, that's cancer. So when you think of mitosis, just think of a copy machine. You're just making cells that are exactly the same for growth, repair, replacement, right? That's the function of mitosis. Now this rhymes, and I remember when I was in school, before I really was focused and really interested in science and biology, mitosis, meiosis, they sounded the same. The cells kind of look the same. I was kind of like, oh yeah, that's one of those things where chromosomes are moving around, right? Well, we're going to analyze the distinct difference between mitosis and meiosis, and hopefully when we come out of this, you're going to understand that mitosis and meiosis are similar, but they have very different purposes. And the distinct purpose that you can see here is sexual reproduction. The purpose of meiosis is to make sperm and egg. This is how cells go from having 2 sets of chromosomes to only 1 set of chromosomes. So when sperm and egg come together, we're back to 2 sets of chromosomes. So the purpose of meiosis is to make sperm and egg. It's not a copy machine. It's not to make new cells to replace or repair or for growth. It's specifically making sperm and egg. And where are sperm and egg made in mammals? Where are sperm made? This is back to health class in middle school. Just say it. Testes, right? It's okay. We're not in middle school. We can say these words. So sperm are made in the testes, and eggs are made in the ovaries, okay? So where does meiosis occur? In the testes and ovaries of mammals. This is biology, so we're not just talking about humans. We're talking about, mammals. So dog, cat, wolf, sperm and egg. So we're taking a cell that has 2 sets of chromosomes, so we call that 2N. Yes? A little bit. We talked about asexual reproduction a little bit with the bacteria. Remember, binary fission was how bacteria just make copies of themselves. That's more like mitosis. So meiosis is when we're going to mix up those chromosomes a little bit and get some variation, so we aren't making exact copies. When sperm and egg come together, we have a totally unique individual that's not exactly like either parent. So yeah. Okay, so you're taking a 2N cell, and after meiosis, we have, meiosis. When we're done with that, we have, I should make that bigger. 1, 2, 3, 4 cells that are, that have only 1 set of chromosomes. So it's just like algebra. We don't write 1N, we just write N, right? Because the 1 we don't need. So we have 1 cell, and then we have 4 cells. So the term that we use for cells that have 2 sets of chromosomes, the term is called diploid. Di meaning 2, so we have 2 sets of chromosomes. And what is the term we use for 1 set then? Do you remember this from your biology classes? haploid, very good. So we have 4 haploid cells. And that kind of makes sense if you think about it, because sperm and egg, especially with sperm, we need lots of them because they have a long ways to travel depending on how fertilization takes place in the organism. Long ways to travel, so we want lots of cells. And these are not the same looking. Those chromosomes are different. We're going to talk about how that happens. So we're taking a diploid cell in the testes and ovaries. There are diploid cells in the testes and ovaries, and they undergo meiosis to make 4 haploid cells. So, for example, in human beings, this process of meiosis occurs throughout a man's life. He's producing sperm his entire life. So he always has fresh sperm every 60 days or so. In females, this process of meiosis occurs before birth. So in females, if you have a 40-year-old female, those eggs underwent meiosis and were hanging out for 40 years. So is there a chance that those eggs might have some damage to the DNA after 40 years of exposures to different things in the environment and just aging? Yes. So that's why we see more chromosomal problems with women that have children later in life, because those eggs are older. And when that spindle starts to pull things apart after fertilization, after when anaphase completes, we see the spindle breaking and dropping off chromosomes, and we have extra chromosomes sometimes in that egg that gets fertilized by sperm, and then you have things like Down syndrome that occurs in women that are older. But there's kind of a point at which we see a big increase in chromosomal abnormalities, and that's in the like 43, 44, 45. But fertility goes down, so that's kind of nature's way of protecting, you know, having a lot of mutated offspring, is fertility goes down as animals age, as mammals age, and the same thing with human beings. Is a woman trying to get pregnant at 43, 44 is really, really difficult, but a woman can get pregnant in her early 40s. We see a lot of, you know, women that are trying to get pregnant, and they are successful in the very early 40s. But again, the risk of chromosomal problems goes up a little bit, not hugely so, but we go from 1 in 10,000 to 1 in 100. So that's a big jump, right? 1 in 10,000 versus 1 in 100, but still 1 in 100, what is that as a percent? What's the percent? If we have 1 in 100 risk for a 40-year-old woman to have a child with a chromosomal problem, what's the percent? 1% chance. If someone said you had a 1% chance of this or that, would that upset you and make you think, oh my gosh, I'm not going to do that? Right? 1% is pretty small. But if you have a room full of babies born to women 40 to 41, and there's 100 babies, one of them is going to have Down syndrome, right? But if there is a slight increase, and they're not quite sure why, there is a slight increase in men who are older who have children, having children with autism, and they're not quite sure why, but of those kids with autism, some have older dads. There's a, for whatever reason, we're not quite sure why that is, but yeah, there's. Boys, there's more fertilization of boys there's more fertilization of egg with male carrying sperm, the Y carrying sperm, because the chromosome for male gender is a little tiny Y chromosome, so those sperm can swim a little faster. They're not carrying that heavy X. But there are more issues with chromosomal problems and miscarriage with male sperm. And we'll talk about that when we get into genetics. So there's an equal chance of fertilization, you know, of a chance of having boy or girl, but we find that more girls are born than boys because of the Y chromosome versus the X chromosome. And we'll get into that with genetics, so we'll follow up on that. Anyway, so moving on. So meiosis then is used for sexual reproduction. So that's the only purpose. It's not for making new cells. It's not for growth, repair. It's not related to cancer. Again, it's for making sperm and egg. Or making the gametes. The term for gametes just means sperm and egg. That's the fancy term for sperm and egg. So it produces haploid gametes, which again is sperm and egg. That's what gametes are. So it only occurs in testes and ovaries. So you have a sperm that has one set of chromosomes, and you have an egg with one set of chromosomes. When that sperm fertilizes that egg, we end up, N plus N is 2N. And we call this a zygote. And that's the first cell that we all came from. So this is the egg. This is the sperm. So when sperm nucleus fused with the egg nucleus of your parents, that formed you. And then from here, it's a copy machine, okay? So forming these two cells happened by meiosis. Once we get back to the 2N, it's mitosis that takes over, that turned you into a fetus that was born and growing from there. So let's take a look at the human nucleus. So all of your body cells have two sets of chromosomes. So chromosome number 1 carries specific information that makes you you. So we can see that we have 23. The 23rd pair is called the sex chromosome. So that's the 23rd pair. So that's going to either be XX if you're a girl, or XY if you're a male. So this is the 23rd pair. So we have 23 different chromosomes, and we have a pair of each. So how many total chromosomes do we have? 46. 46 chromosomes. So in all of your body cells, so if I look at your hair, your skin, your bone, any body cell, they're going to have 46 chromosomes. So we have 46 in all of our body cells. But then in the testes and ovaries, there's a process called meiosis that takes a 2N cell, divides it, and forms multiple haploid cells with only one of these chromosomes. So a homologous chromosome is a pair of chromosomes that share similar characteristics, but they're not exactly the same, because one of these chromosomes came from sperm and the other one came from egg. So that's why we have a set of each, because when this happened, 23 chromosomes, chromosomes 1 through 23, chromosomes 1 through 23, we had a pair. So you have this. So if I look at sperm or egg, I would just take away one of each of these, and that's what the sperm and egg had, was only one of these chromosomes. So whenever a cell is getting ready to divide, whether it's by mitosis or meiosis, the chromosome that we have, so here's one chromosome, remember it replicates and forms a sister chromatid, so we can begin this process of cell division. So this is a replicated chromosome. So when you have two replicated chromosomes, when this one replicates and this one replicates, we end up with these two replicated chromosomes. We call these a homologous pair because they're similar, but they're not the same. Okay, they're similar. They carry similar information, but one came from mom, one came from dad. So they're not exactly the same, because mom and dad sure don't look alike, right? So they're slightly different, but they are human beings. They both have 2 arms, 2 eyes, 2 legs, right? So they're similar, but they're not exactly the same. So there's different traits on these chromosomes that we're going to study in the next unit, looking at genetics and heredity. But we're not, right now we're just focusing on where did these chromosomes come from that we have. So normally chromosomes are single, chromosomes, one strand of coiled up DNA. Then they enter the S phase of interphase, right? And they replicate, they make a copy of themselves, and they're connected by the centromere. So this happens in sperm and egg, so we have these X-like structures. And then they separate in anaphase, and then we're back to this again, right? So we're going to talk about this process. Again, these chromosomes, these homologous chromosomes, one came from mom, one came from dad, and they're very similar. So this is a homologous chromosome. But chromosome number 2 and chromosome number 1 are not homologous because they carry different information. So when we're looking at homologous, we're looking at a pair that are the same chromosome but from a different organism. All right, so if we look at what occurs on the chromosomes, each little banded thing represents a different trait. And variants of those traits we call alleles. So an allele might be, let's say, widow's peak. Widow's peak is if you pull your hairline back and you see a point come down. I'm trying to look across the room of people who I can see have a widow's peak. You have a widow's peak. Yeah, if you pull your hair back. Can you see how she has that point that comes down in her forehead? That's a dominant trait. Now you can all look in the mirror when you get home. So the trait is widow's peak. If you have it, we give you, we give you big W for widow's peak. If you don't have it, we give you little w. So I'll just put a line over the top. So little w. So that big W means you have it, little w means you don't have it. So if I look at these chromosomes, what if dad had a widow's peak? Then we would have big W. And what if mom didn't have a widow's peak? Then it would be little w. So each of these traits are represented on these homologous chromosomes. And this is me. This is what I'm going to look like. Am I going to have a widow's peak? I would, because in this case, this trait, the big W dominates over the little w. And if you have big W, you're going to have a widow's peak. What if, so I'll put a little line meaning this is little w. What if you have a parent, your dad does not have a widow's peak, your mom does not have a widow's peak, you're going to have two little w's. Is it possible for you to have a widow's peak? No. So we call this homozygous, meaning that the alleles are the same for that trait. We call this heterozygous, meaning you have different alleles for that trait. And it depends if it's dominant, if it's going to override this, the dominant allele, depending on the type of heredity. And again, this is the next topic, so I don't want to get ahead of myself. I just want you to understand what alleles are. Alleles are forms of a trait. So you're either dominant or recessive for a trait. And depending how those two alleles come together, this is called homozygous recessive, this is called heterozygous. All right, so identical alleles, if they're the same, we say that you are homozygous for that trait. And if they're different, we say you're heterozygous. So homozygous could be either big W, if you have two parents with widow peaks, or if neither parent has a widow's peak, then you would say you are little w. But if you're heterozygous, it means you have one of each of the alleles. So again, alleles are variants of a trait. So in this case, it would be big W and little w, if you're looking at widow's peak. Okay, so now we're going to look at the processes of meiosis. And I really feel like the process of meiosis is best seen in looking at an animation of this process. So in our folder in Blackboard is an animation that we're going to step through. And as we go through, what is the problem here? There we go. These are the two animations I want you to walk through and look at the differences and similarities between meiosis and mitosis. So after watching the animation then, again, key concept that you should pull from the animation is that those homologous chromosomes pair up in prophase 1, and that's called synapsis. When they're together side by side, we call them a tetrad. Tetrad means 4. And then those homologous pairs separate. In what stage of meiosis 1 do they separate? Yep, in anaphase 1. So anaphase 1, the two homologous chromosomes separate, and then we have a duplicated chromosome from each pair in the cells. And this tetrad is when I take one chromosome from mom, and then I take the other chromosome from dad, and we're just bringing them close together like that. That's called a tetrad. And that process, remember, synapsis just means coming together. And when we have the two chromosomes really close together, can you see that there's 4 arms to it, right? There's 4 sister chromatids that are really close together. That's where the word tetra. Tetra means 4. So this is called a tetrad. And again, this is formed in prophase 1. So these 3 things happen in prophase 1. So then they line up in metaphase 1, and then meiosis 2, the sister chromatids, the sister chromatids of these tetrads, or of these homologous chromosomes, separate. So sister chromatids separate. We have 4 daughter cells that are formed, and only a single chromatid exists. So we look at meiosis 1 here, is at the bottom. Key thing is we're separating homologous pairs, and in meiosis 2 we're separating sister chromatids. So here's my chromosome, or my cell, sorry, in the ovary or testes that's going to form sperm or egg. They replicate, they form tetrads, they line up on the plate. So this is meiosis 1. They separate, and we have 2 cells that are haploid, right? After meiosis 1, we have 2 haploid cells. Then meiosis 2, the sister chromatids separate by undergoing prophase, metaphase, anaphase, telophase, and we have 4 cells here that are all haploid. So again, we had a 2N cell at the beginning. We have 2 haploid cells after meiosis 1, 4 haploid cells after meiosis 2. So that crossing over, again, remember, is when we're exchanging genetic information. And that introduces genetic variants. Question? Yes, it does develop, redevelop at the end of meiosis 1. The only thing that doesn't happen before meiosis 2 is we do not see DNA replication in between meiosis 1 and meiosis 2. So we have just these chromosomes that were already formed by replication here before meiosis 1, but we don't see it before meiosis 2. So this is crossing over. These are showing, yes? It's very brief. It's very brief. And then it just goes right into meiosis 2. Yeah, there is no interphase where it's like hanging out for a very long time, but it does, we do have the growth phase and, but not the replication, like I said, of the S phase. Okay, so these are alleles for a trait, big B and little b, so they're just showing, let's say, that this is on dad and this is on mom. With this crossing over that occurs, we see an exchange of these alleles. And now the daughter chromosomes that resulted from crossing over, I don't know what the problem is here, it won't let me do that, we see different chromosomes now. They have different information. Instead of A, B, C, D, this one is A, B, and it's got a little bit of this chromosome piece after crossing over, so all 4 of them are distinctly different. So crossing over is all about genetic variability. And these are all different sperm and different egg that's going to have different information at fertilization. Another term that gives variability to sperm and egg is a term called independent assortment, which means how these chromosomes line up on the metaphase plate is variable. For example, all of mom's chromosomes could line up on one side, all of dad's on the other, or we could have a little mixture. I mean, look at all these combinations. With just 3 chromosomes, we could have 8 different combinations of how they line up. So when they separate, each cell is going to look a little different. So now picture, what, 23 different chromosomes. That's a lot of variable combinations of how they line up on the metaphase plate. So when you hear the word independent assortment, we're just talking about how they line up on the metaphase plate, and that is totally random. And we don't know if dad or mom's chromosomes are going to line up on what side. Okay, and another thing is fertilization. Which one is going to be fertilized? Which sperm is going to fertilize what egg? Again, more variation. So with crossing over, if crossing over only occurred one time, we have 4 different chromatids, 23 different chromosomes, we have sperm and egg, we have this many possible different zygotes that can occur by crossing over. That's, I wouldn't want to begin to say it. It's a really, really big number, right? So it's amazing that we do see brothers and sisters that look so similar, isn't it? When you look at brothers and sisters, like, and you say, oh my gosh, you look like twins, and they're born 2 years apart. It's amazing because there is so much variability that happens as a result of meiosis. So again, sexual reproduction is important because we need environmental adaptation over long periods of time. So we look at these moths. If you have trees that have dark bark, which ones are going to have an advantage here? If you have trees with dark bark, which ones are going to be eaten by birds? These, where these with the dark color are going to survive. And that did happen way back. There's a story about that in the Industrial Revolution. There's a lot of smoke and coal that coated the trees and made them very dark, and those moths survived that had the darker color. These guys stick out and they're more likely to be eaten. So when we come back next time, we're going to compare meiosis to mitosis.